Hello, everyone, and welcome to today's broadcast, Human and Environmental Microbial Health, a Global Perspective. It is presented by Jack Gilbert, PhD, Faculty Director of the Microbiome Center and a professor in the Department of Surgery at the University of Chicago. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Illumina. For more information about our sponsor, visit www.illumina.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, just click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Gilbert. I will now turn the presentation over to him.
Hi, thank you very much. It's a great pleasure to be here. So today I'm going to be talking about how human and environmental microbes, that's bacteria, fungi, viruses, in all the ecosystems of the planet, including our own bodies, how they interact with the world around us, and how they interact with each other to shape the health of both our environments, be it agricultural environments or our natural environments, and our own human health in medical practice. And I'd like to start by giving one of my favorite quotes by one of my colleagues from University of British Columbia, Julian Davies, who said that once the diversity of the microbial world is catalogued, it will make astronomy look like a pitiful science. And I love this quote because not only does it claim numerical superiority for the microbes, but it stipulates a very obvious trend towards understanding the biological phenomena that is microbial diversity. This is a microbial planet, and we need to understand how the microbes manage this planet if we're to ever play a significant role in manipulating the planet and managing it effectively as stewards. So there are currently, I think, 1 times 10 to the 30 bacterial cells in the ocean and 1 times 10 to the 24 stars in the known universe. So one would argue that there is definitely a lot more microbial life than there are stars to look at, which is always makes me feel better. So to understand this concept of microbial diversity, in 2010, we launched a project called the Earth Microbiome Project. This is a collaboration initially between myself, Rob Knight at the University of California, San Diego, and Janet Jansen at the Pacific Northwest National Laboratory. And we wanted to put in place a program that would allow for continuous and integrated and standardized monitoring of microbial life on this planet by partnering and collaborating in open data science across the globe. The study started by basically asking people if they had spare samples in their freezers, which they'd catalogued maybe nutrient concentrations from, or they looked at the health of a person but hadn't sequenced the bacteria in their stool. And so we asked them if they wouldn't mind donating those samples to the project, and it rapidly span out of control from there. As it stands, we published the first paper at the end of last year. And that paper had around 28,000 samples that have been processed and analyzed using standardized approaches to understand the microbes that are present there. And currently we have well over 100,000 samples that have been processed for 60-ness ribosomal RNA amplicon sequencing using the Illumina sequencing platform in a standardized way that has standardized both DNA extraction, the PCR approach used for amplifying that gene, and the sequencing and analysis. All of this data, over 10 billion 60-ness reads, 1.2 million unique sequence fragments, are publicly available for anybody to use, download, and publish on through the CHITA database, the QIITA, which you can access through earthmicrobiome.org. And that means that you could not only take that data and do a large meta-analysis of any environmental systems that are currently available, but you could also generate your data for your samples through the Earth Microbiome Project standard protocols, which are also available on the website, and then compare your sample environment to thousands or tens of thousands of other ecosystems across the globe, which can significantly enable contextualization of your sampling set in response to other people's data. The Earth Microbiome Project covers many different environments. Here I show some maps where we catalog the different animal and plant-associated microbes and their geolocation. Obviously, you can see there are lots of animal-associated samples, not many plant-associated samples, and they're poorly geographically distributed. But we have gaps to fill, and we're fully aware of that. This is the map of just the first 28,000 samples, so we've already started to plug some of those gaps. And then I catalog saline and non-saline environments. Obviously, the saline environments are the marine system, the oceans, but also inland lakes that have salt or soils that are salty. And then we have non-saline environments, which obviously soils and lakes and rivers that have fresh water. And we've done a pretty good job at capturing some of those environments. And why have I divided these into animals and plants and saline and non-saline? Well, that's because at the Earth Microbiome Project, we needed to be able to catalog environments in ways that would allow us to understand where those samples had come from. And we did that by trying to understand the factors which significantly explained the variation in microbial ecosystems across the planet, right? So if we look at the key factor which appears to describe the bacterial communities on the planet and archaeal communities on the planet, it's whether the microbes are free living or whether they are associated with a host. And those animals or bacteria, that are associated with a host, for example, often have a smaller genome and they're very well adapted to that host environment. Whereas those that are free living often have larger genomes and they're more adaptable to changing ecosystems. And then we divide the free living organisms down into whether they belong to a saline or a non-saline environment. And then we have further ontologies that go deeper into those kind of environmental processes. The hosts associated are divided between animals and plants. And again, each one of those ecosystems is further divided. And if you look at the bottom left corner here, you can see the diversity of those environments. So in the host-associated samples, the most diverse environments are often the rhizosphere of the plants, which are obviously buried deep in a very complex system, soil, which just so happens to be the most diverse free living environment as well. So we can see paralogs between those systems. Obviously, the least diverse environments are things like animal corpses, or the animal body, so skin and things like that. And then the least diverse free living environments are things like water from oceans, which are very selective for certain organisms. And then on the top left-hand corner here, you can see the PCOA plot, where every single dot here is an entire microbial ecosystem. And when the dots are closer together, they're more similar. The further apart, they're more dissimilar. And you can see that there's a continuous distribution of microbial ecosystem properties. It's not discrete clusterings. There's a universal almost construct that allows for the huge amount of diversity to be represented. And then one of the key things that we found was that ecosystems that had a lot of microbial organisms that were very diverse, as you can see on the left-hand side of this graph, where samples have lots and lots of different types of microbial phyla, compared to ecosystems which have very low diversity over here on the left, where the organisms have the samples have very few microbial phyla. We found that for the vast majority of environments, those environments that were very low diversity contained microbes that were found in very highly diverse environments. So what we call this microbial nestedness. I like to think of it almost like osmosis, right? So environments that are highly diverse are constantly leaking in microbial communities, microbial taxa, to environments that are very selective. And only those organisms which can survive in that selective environment will survive, and hence you get low diversity in those selective ecosystems. But the organisms come from very highly diverse environments. And that's a very important concept, because it means there's distribution of microbial life across the world, both for soils and oceans, but also for our own bodies. And understanding where those organisms come from and the forces which select for them to be become colonized in our bodies, for example, are incredibly important to understand. When we actually look at microbes at different levels of genetic phylogeny, so whether they are a genus or a species or a strain, a subspecies, we can see that the distribution of organisms is very, what I would like to call phylogenetically restricted. So, for example, here on this top graph, we are seeing genera which are highly distributed across different environments. So some of these genera can be found in human bodies, animal bodies, in soils, in plants, in ocean samples. So the genera is found, the genus of bacteria, for example, is found in many, many different ecosystems around the world, right? But when we go down to the subspecies level, down here on the bottom, these are organisms which are strain level. So they often have a few nucleotide polymorphisms different in their genome compared to other species, other members of the same species. You can see some of those strains are highly resolved to certain ecosystems. So this big green blob in the middle, we can see that big green blob is a microbial strain that is very highly conserved in plants. And then in the little bit of red at the top, it's also found in the soil that surrounds those plants. So we have this organism which is highly adapted to that particular environment. But fellow members of that species can be found in various environments around the world, suggesting that you have this evolutionary selective pressure which creates these unique organisms that are super adapted to particular environments. For a bit of fun, we wanted to know whether we could take these data and model some of the microbial ecosystem processes that these microbes provide. For that, we did a fun exercise. So here in this rotating globe, you can see the distribution of soil moisture at a very coarse resolution across the planet Earth. And we have this property, and we have temperature and nitrate concentrations and carbon concentrations and various other properties of soil across the planet, based on modeling. So these are predicted variant measures. But we can use that. We can tie those ecosystem properties to the microbial communities that are present there, just based on correlation. So, if we have this particular soil moisture, this particular pH, this particular temperature, then you see that these microbes are associated with those particular properties by statistical correlation. You can then extrapolate the presence of microbial proportions across the planet, as we've done here for alpha proteobacteria. So we've taken an entire class of microbes and said, how well are they distributed across the planet based on the environmental constraints that we see across the ecosystems of these soil environments. So now we've used the environmental parameters to predict who's there. We can also say, well, what are those organisms capable of doing? So we can compare who's there to genomes in the genome database that tell us what those organisms are capable of doing, what their genomic functional potential is. And we can use that information then to combine those properties together to predict the relative proportions of their genes. So here I've represented cellulobiosides across the planet using the genetic information from the alpha proteobacteria. Once we know what they're capable of doing, we may even be able to predict what their ability is to turn over certain metabolites in the environment. So we've done that here with carbon dioxide, predicting the ability of those microbes that we're able to understand in the soil to turn over CO2, to either synthesize it or consume it as a relative proportion. But remember, these are all predictions based on predictions, based on predictions on predictions. So it's a bit of fun, but myself and my colleagues can use this information to develop particular hypotheses, which could then be validated in either observations of different environments or in experiments around the world. So take them for what you will. We've used these kinds of modeling in collaboration with Noah Fiera and various other colleagues around the world to try and understand the microbial ecosystem properties and how they shape or are shaped by the environmental properties. So this work, also collaborating with Joshua Ladau, was trying to understand if we could map the distribution of microbes in what would have been the Great Prairie of central of the Midwest in the North American continent in the 19th century. So what you can see here is a map that shows the distribution of that Great Prairie before it was eradicated by agricultural revolution. And we've mapped onto that the distribution of Verrucomicrobia, a particular phyla of bacteria, that are incredibly abundant in the central area of that prairie. And the blue in the periphery shows that particular phylum becomes very rare on the outskirts of the prairie distribution. How have we done this? Well, we snuck into graveyards and railways that have been there since the Civil War. And we looked at the bacterial communities using metagenomics on the Illumina sequencing platform that would allow us to determine who was there and what functions they were capable of achieving. And then we basically mapped the distribution of those microbes in those discrete locations to climate and said, well, okay, well, if I extrapolate and interpolate between these point samples, can I predict what the distribution of these organisms would have been had the prairie been contiguous across its entire extent back in the 1840s? So here, that's how we got to this distribution of Verrucomicrobia. Why do we think Verrucomicrobia is important? Well, it's very hard to culture, so we don't know a lot about them. And they appear to flourish when you have a lot of carbon, but not very much nitrogen, as you do in prairie environments, such as those found in graveyards and railway sidings from the Civil War. Before the introduction of nitrogen from agricultural fertilization experiments, right? So when we started to take over the Great Prairie and start to plant crops and then fertilize them with inorganic and organic nitrogen, we started to eradicate the likelihood that Verrucomicrobia could survive. And we can see that the Verrucomicrobia significantly affect the functional potential of these ecosystems. If we look at the graph on the right here, which shows, again, the distribution of microbial functional diversity across the Great Prairie extent as it would have been in 1840, we can see there's very low functional diversity right in the middle where the Verrucomicrobia would have been highest. So the Verrucomicrobia were dominating and they weren't very functionally diverse, which suggests that the prairie grasses and the Verrucomicrobia were in a very tight partnership. So if you wanted to restore the Great Prairie in any environment, you'd have to make sure that you got those Verrucomicrobia which were in tight metabolic or relationship style partnership with those grasses to allow them to flourish in that environment. The soil, the microbes, and the plants are in a almost symbiosis with each other that allows for the development of a stable ecosystem. We wanted to know what this particular Verrucomicrobia population actually was and why it was so dominant. So we took these genomes and these metagenomes, and we actually assembled genomes of these Verrucomicrobia from that population. We isolated a strain that we call DA101, which is a genotype, again in collaboration with Noah Fiera. This strain of Verrucomicrobia was then mapped to metagenomic reads from different projects around the world, including, if you look at this, the bottom middle graph, from the grasslands that are non-fertilized in Central Park. And you can see that this one particular species of Verrucomicrobia, which has only been categorized based on an assembled genome from metagenomic data, was actually incredibly abundant in grasslands all around the world in many, many different environmental studies, suggesting it's incredibly well distributed, which is further support for our conclusions about the distribution potential of microbes across the planet. In this study, which we worked on with colleagues from a number of different universities, but particularly Zhejiang University down in Hangzhou in central China, we analyzed the microbial diversity based on the bacterial 16S and archaeal 16S and fungal properties based on the ITS fragment through amplicon sequencing, and identified the microbial diversity across soil samples from forests all the way down the coastal regions and central regions of China. The very top environments in blue and pink there are very dry, mostly dry soils, non-tropical environments, going all the way down to the very bottom in orange with very tropical environments. And the soil properties are therefore under different climatic environments. So what we wanted to do here was provide a map of the microbial diversity of these environments that could undergo significant shifts in climate properties over the next 100 years or so to determine if they are changing. So we provide a baseline for these systems. Using this data of archaea, bacteria, and fungi, we produced network mapping and network associations. So here the dots are colored by whether they are bacterial taxa, eukaryotic taxa, or archaeal taxa, and every single dot is a microbial organism. And its relative abundance was then correlated with the relative proportions of all of the other microbes in the system. And there are here nearly 65,000 statistically significant, highly strong partnerships, strong, significant correlations between the relative proportions of these groups, representing some 3,900 bacterial, archaeal, or fungal taxa, OTUs. 90% of these interactions that you can see here are universally distributed across all of the climate regions that we mapped in this study, whereas only 10% of them are region-specific, i.e. only found in one of these particular climate regions. So if they are climate region-specifics, those 10%, they're going to be incredibly important to understand what those associations actually mean for the microbial ecosystem. Because if that climate region shifts to a different climate type, those particular associations are going to be under threat. One way we can see this is by mapping the strength of the associations across the gradient. So up here you can see when we map that as a gradient statistic for the topological strength of those networks, the environments in the north, in the far north, have very strong associations, statistical associations, very strong correlations between different microbes, but not many correlations. So they're very strong, but fewer associations. Whereas down in the south, the associations are weaker, but they have a lot more associations, which is a very interesting phenomenon. What we hypothesize that this is due to is the fact that in the north, where the soil is mostly dry, the microbes are highly dependent upon the associations they have within their own biofilm on a particular granule of soil. Now, if it was to rain there, then those microbes would be able to then exchange metabolites with the biofilms and the other bacteria and the other fungi on other soil grains. But for the most part, when the soil is dry, they do not have that potential. So they become incredibly dependent upon the microbes within their own biofilm. Whereas in the south, where there's lots of soil moisture and therefore lots of opportunity to exchange metabolites between organisms that are not within or on your own soil grain, you have a much greater promiscuity of metabolic relationships, which provides you with potentially a lot more stability or resilience to change, because you're not so reliant on one association or two associations with organisms in your biofilm. We're suggesting that the communities in the north that are those associations that are climate specific in the north are going to be under much greater threat of under a changing climate. And we can use this information to help growers, so especially low input, smallholder, tropical agricultural groups, which the vast majority are women, that support some 900 million of the world's poorest people. By understanding how the ecosystem interactions between microbes is affected by, say, different agricultural practices and climates, we should be able to make better predictions about management strategies for ensuring that those farms remain productive and can become even more productive, and therefore provide the women that run those farms with greater revenue, which could have significant social impact in some of these poor countries. For example, here we can see that increases in nitrogen addition on the x-axis on the graph on the left has a significant correlation to decreased microbial diversity, causing or leading to significant selection pressure for specific types of bacteria that can survive under increased nitrogen potential. So we need to be able to understand that relationship, just as we did with the Verruco microbia. This is obviously interrelated to human health. The nutrition that we would get from those farms significantly alters our microbial cells, but also plays a very strong role in the development of our bodies. And our bodies are microbial ecosystems. There are some 35 trillion microbial organisms on our bodies or living in our bodies. That's almost comparable to our number of human cells, which is about the same, 35 trillion or so. So we're at least half human. But we have also around 27,000 human genes, but there are approximately 3 to 5 million bacterial genes, especially living inside our body. So that genetic potential for microbial life in our body is huge. And therefore, it's no surprise that our body has become reliant upon some of the metabolic chemical relationships that the microbes provide to our body for its adaptation, for its development, and for its proper functioning. And we understand this by using a study population we call microbiome-wide association studies. These are akin to genome-wide association studies, where you would look for a gene or an allele in a human population that associates with a particular disease or health state. We do the same with microbes. We just look at the genes or the metabolites or the organisms which are associated with particular disease traits or functional traits in a human population. And mostly this is due to the metabolic relationships of those microbes. So microbes are little chemical factories, right? They produce huge arrays of chemistry which are sensed or used by our body for particular purposes, such as they synthesize amino acids, which are essential for our physiological functioning. They synthesize chemicals which they use to communicate, which also help our body cells to communicate. And our body cells become reliant upon those chemicals. They can even break down things like acetaminophen, so drugs, and they can produce, some of them, negative chemical byproducts from that metabolism, such as acetaminophen sulfate, which can be toxic to our liver if you're on a long, chronic use of that drug as a painkiller. That's very important to understand. They can also break down some of the chemicals our body produces, like bile acids. The secondary bile acids, some of the microbes in our gut synthesize from their catabolism of bile acids, can be incredibly important in keeping some of the pathogens that invade our bodies in check. Specifically, things like Clostridium difficile. Some of the secondary bile acids bacteria synthesized in our gut can keep Clostridium difficile, or C. diff, sporulated, so it becomes a spore and it stays a spore, and therefore it doesn't infect us. The important thing about this to understand is that everything touches everything. One of my good friends, John Alverdi, taught me that. This is a quote from Jorge Luis Borges. But this study of microbial ecology in all environments is incredibly important for human health. And we are now starting to understand how the microbial ecology of our bodies affects our health, our wellness, and leads to disease when it breaks down. So we now treat the body as an ecosystem. And that's the reason we have many microbial ecologists like myself turning up in departments of medicine across the country. Microbes can affect your life in many ways. I already talked about the Tylenol story, right? So they can break down acetaminophen, which is the active ingredient of drugs like Tylenol, and create products that are toxic for our liver. But the microbes in our skin can also break down our sweat, and they use the sweat as a metabolic product, and a byproduct is often smelly chemicals, aromatic compounds and volatile compounds, which are sensed by insects like mosquitoes, which will then use that signal to hone in on a certain part of our body and bite us. if we can interrupt that signal, we may be able to stop some of the microbes from some of the mosquito-borne diseases, for example, from overcoming our populations. We also, down in the left corner, you can see a picture of two flies humping, which is also the name of my new album. That's a joke. But we found out that microbes in animals can play a very significant role in modulating behavior, potentially through many routes, such as the gut-brain axis, where they synthesize neurotransmitters, which can bind to our vagus nerve and alternate signals to our brain. But they can also microbes can affect our inflammatory status, our inflammation, which can affect how animals behave. And we've shown that animal, especially fly, reproduction selection, so what their mate preference, can be significantly affected by the bacteria present in their guts. We think that just like in any ecosystem, when we remove certain species of microbe from that ecosystem, it's going to have an effect. So if I remove all the trees from the Amazon jungle, for example, it's going to affect the hydrology of that ecosystem, affecting local climate, affecting the animals that can survive there. So we would class the trees as a keystone species. Many of my colleagues, such as Martin Blazer, have started to understand that the missing organisms in our bodies that have been wiped out due to over excessive antibiotic use or poor diets or living indoors have significantly altered the microbial ecosystems in our bodies, eradicating some of those keystone bacteria that make our bodies work. And this has led to an explosion of certain disease types. So we hypothesize. And there are currently over 400 ongoing or completed clinical trials on clinicaltrials.gov that are there and where the microbiome is being investigated as a potential cause or microbial change is being investigated as a potential biomarker for that disease. Everything from cancer and inflammatory bowel disease all the way through to liver diseases, sinusitis, influenza. And then, people are trying to understand how things like exercise and smoking and drinking alcohol are affecting the microbial ecosystem in our body and maybe feeding back that damage on the microbes into consequences for our own health. One of the keystone studies demonstrating that microbial ecosystem properties could play a role in treating disease was the use of an infusion of donor feces, or what we'd call a fecal microbiome transplant, from a patient with recurrent Clostridium difficile, from a patient who's healthy into a patient with recurrent Clostridium difficile infection. So here, if we give those patients who have recurrent C. diff infection vancomycin, which is the standard treatment, an antibiotic, therefore, we get about 30% success rate. So 70% of people are still very sick and still getting recurrent C. difficile infection. If we give them an infusion of donor feces from a healthy donor, we get almost a 94% reduction in the likelihood of that disease progression. And this is not a new therapy. The Chinese used to call this yellow soup, where you'd feed the stool from a healthy baby to somebody with chronic diarrhea, and it was supposed to be highly effective. But we are now weaponizing this, and groups out there, non-for-profit organizations who provide healthy stool for this treatment, have now fueled almost 25,000 fecal microbiome transplants for C. diff patients around the country, with an estimated cure rate of approximately 90%. We know that the microbial ecosystems of our body affect our health, but we're also finding out that microbial exposure that we get in the natural world can affect our health. One study that we did with Anne Sperling and Carol Ober at the University of Chicago in collaboration with Donato Vercelli at the University of Arizona was to try and understand how the microbial exposure that children get on farms could be influencing their immunological health. This comes about from some extremely important work from Erika von Mutius over in Germany at the University of Munich, which has predicated this in German populations. But we looked at it in our own unique US populations of the Amish and the Hutterites. The Amish and the Hutterites come originally from Eastern Europe over a couple of thousand years ago, resided for the last 1,000 years in Northern Europe and Central Europe, and in the 19th century emigrated to the United States where they set up shop, mostly the Amish in Ohio, Pennsylvania, and Indiana, and the Hutterites up in Dakota. They both live very similar lives in the fact they reject technology. So they don't have iPads. I can take my children to this farm to show them that they can survive without an iPad. Children do it. But they also consume a lot of fermented foods, drink a lot of fermented milk products, consume unpasteurized milk and eggs. And they are living a very wholesome food lifestyle in a traditional farming environment, German food lifestyle. The main difference between the Amish and the Hutterites is that the Amish live on their farms. So children in the morning and before school and then after school are working with the animals on the farm. And they pretty much do this from birth when they're strapped to their mother or father as infants. Whereas the Hutterites, the farming environment is actually distant from the home commune. And so the children living in the home never get real exposure to the farming environment, at least not until they grow older and start to work there over the age of 14. We think that critical difference in the early life exposure that children get has a significant consequence. One of the major clinical differences between the Amish and the Hutterites is that the Amish have about 4% asthma in their population, 3 to 4%, significantly lower, about half of the US national average, whereas the Hutterite population can have up to 20 to 30%, an average of about 25% asthma in their population, suggesting either a genetic basis, which has been very hard to track down, or a difference in environmental exposure, which we predicate is due to this difference in early life exposure. When we look at the children's immune systems in these two different environments, we can see that there are telltale signs of differences in microbial exposure. So the blue boxes here are the Amish kids, and the red boxes are the Hutterite kids. The top left box represents neutrophils, which are, for those of you not au fait with immunological cells, are like little scouts that run around the body looking for foreign objects. And when they latch onto a foreign object, they send out a signal which activates the immune system to come and clear it out with macrophages and eosinophils, which promote inflammation. The neutrophil will die in the process of that. Interestingly, we see a lot more, statistically significantly more, neutrophils in the Amish population, suggesting that they have a lot more exposure to foreign objects, potentially from their interaction as children with cows and pigs and horses, compared to the Hutterite children. The Hutterite children have a lot more eosinophils in the top middle box there, which are pro-inflammatory immune cells, suggesting they have a greater inflammatory systemic inflammation and therefore a greater likelihood of having a triggered inflammation. The most interesting thing to me is the middle three boxes, where we see cell surface markers present on neutrophils being significantly elevated in the Hutterite community. So the Hutterite community, the Hutterite children have less neutrophils, but their neutrophils have a lot more of these cell surface markers. These cell surface markers appear when the neutrophils get old, i.e. when they're 2 or 3 days old, they can get an elevated level of these cell surface markers. They're not interacting with anything in the bloodstream because they're not seeing a lot of foreign objects. They stay around in circulation for longer. These cell surface markers make the neutrophils more sticky, and when they stick to other cells or to each other, they can promote a greater degree of inflammation, a greater degree of eosinophil recruitment, and therefore greater inflammation and a heightened likelihood of asthmatic attack. To prove this, Donata Vicelli in our group took, at the University of Arizona, took germ-free mice and specific pathogen-free mice, and she sensitized those mice to an allergen, valbumin. So when the mice got a dose of valbumin, they would have an eosinophilic response. And then what we did is we exposed those mice intranasally, so made them breathe in dust from the bedrooms of either Amish or Hutterite children. And so on the graph on the left here, you can see that when we look at eosinophils, for example, when we give the mice just PBS in gray, just a phosphate buffer, they make them inhale that, they have no allergic response. When we give them a valbumin, they have a significant allergic response, a significant increase in eosinophil recruitment. And when we give the mice Amish dust intranasally, and then we give them the valbumin, it significantly reduces their eosinophilia. So they have no inflammatory or very low inflammatory response to the valvumin, the thing they're allergic to. It also significantly increases, here in blue, the number of neutrophils. And that is exactly what we saw in the Amish children. When we give the mouse Hutterite dust, or dust from the Hutterite children's bedrooms, and then we expose them to a valvumin, significant degree of inflammation, heightened elevated eosinophilia, and a significant decrease compared to the Amish children in the number of neutrophils. Exactly what we saw in the Hutterite kids. And this is a nice proof of principle that the Amish dust contains something which appears to desensitize this animal model to particular allergens. Microbes also play a significant role in obesity. I love this story because one of my colleagues lost a lot of weight by going on a traditional Chinese whole grain diet, where he only ate whole grains for about 9 months. And he checked his microbiome out before and after he lost all the weight. And there was one organism, Enterobacter cloacaceae B29, which was significantly more, which when cultured up and added to a mouse, increased the mouse's fat content significantly. This picture is not exactly from that study, but it's very indicative of what happens to the mice when you add Enterobacter cloacaceae B29 into their guts. This happened in germ-free mice, so both mice are receiving the same calorific intake. They're having the same exercise regimen. The only difference is the presence of this organism. So you've got choice. This organism is either increasing the amount of nutrients and energy these mice are getting from their diet, or maybe its metabolites are affecting how the mouse's body processes energy. And in work we did with Vanessa Leone and Eugene Chang at the University of Chicago, we demonstrated that a high-fat diet, represented here by HF, significantly alters the microbial community in the gut, promoting the abundance of bacteria which produce metabolites such as hydrogen sulfide, therefore significantly increasing the amount of hydrogen sulfide in the blood of the mice that we put on this diet. And that appears to be influencing the circadian rhythm genes in the liver. So what we hypothesize is happening is that the high-fat diet will actually influence the metabolic rate, especially over time, of microbes in the gut, selecting for organisms which produce metabolites which actually affect the circadian rhythm of your internal organs, disrupting your metabolism and therefore leading to elevated weight gain. It does this potentially by decoupling the circadian rhythms of your organs from your brain. Something very similar to that happens when you are jet lagged or when you're working nights, when your brain is sensing time differently to your body's organs. And in that system, again, you are very much more likely to put on more weight for the same calorific intake than you would do normally. So there's a similar trajectory inside the body that we think is incredibly important to understand if we want to alleviate weight gain. We've also looked at microbial mapping in different environments. You are, as I say, you're a microbial ecosystem, and you're releasing 35 million microbial cells into your immediate vicinity every hour. So in a study we did in home environments back in about 2013, we were able to map the movement of microbes between people and their home environment. So for example, here you can see noses, hands, and feet, microbial distribution or movement over time from people's feet to the floors of their home and their floors back to their feet, and from their hands to surfaces they interact with their hands, such as doorknobs and light switches, and then the movement of microbes between people, between their hands and their noses. And we see a significant degree of movement. When we add dogs into that home, for example, here we've added 3 dogs, we see a significant increase in the density of microbial movement between people, especially, and a significant increase in the degree of microbial sharing between people and their environment. Interestingly, in another study performed by my colleague Rob Knight, they looked at couples with and without dogs and found that couples with dogs actually share more microbial similarity between each other than couples without dogs, suggesting that there's an important dogs themselves act as an important microbial conduit, which was further proven by this study. My wife took that to heart and made me rescue a dog. This is Captain Bo Digley, who's now a very famous animal in his own right. We also found out that we could use microbial signals left behind in homes to actually track where people have been and what they were doing. So in this particular study, we had 3 people, and person 1 is a 25-year-old man living with his parents. Person 2 in orange is his father, and person 3 in yellow is his mother. And then there are 3 dogs in blue. And what you can immediately see is if you look at the kitchen counter here, the x-axis is time over a period of about 2 weeks. And the y-axis is the percentage of bacteria we found on the kitchen counter that originated from one of those 6 occupants, the 3 humans and the 3 dogs. You can immediately see this is a very gender imbalanced home because nearly 100% of the bacteria found on the kitchen counter come from the mother in nearly all cases. Apart from the end point, when she went away to stay with her sister for a couple of days, and the father's microbiome in orange then explodes onto the surface of the kitchen counter as he desperately fends for himself. We can also see that there are lots of dog bacteria on the floors, which makes sense because they're physically interacting with the floors. There are also dog bacteria on light switches and doorknobs, not because we have dexterous dogs, but because you will distribute bacteria when you interact with Fluffy onto those surfaces as you carry them on your hands. So there is continuous mapping or movement of microbes between us. It just so happens that very occasionally you'll pick up a microbe which has learnt to evade your host defenses and contaminate you and make you sick. And so with this continuous distribution, occasionally you get these hijackers of microbial pathogens which hitch a ride with that free movement and will infect you. So it's important to maintain hand washing, but it's also impossible to prevent the distribution of microbes because it's just so pervasive. And we found this out in surgical environments. In my last slide here, we've done studies looking at surgical infections and demonstrated that for the most part, the reason microbes in our body become pathogenic during surgery is due to stress. So on the whole, surgical infections appear to not necessarily come from the environment, which has a near constant distribution of microbes from people in the surgical theatre, but they nearly always originate inside the patient themselves, in their gut. And the very act of cutting somebody open, which exposes gut bacteria to oxygen, and then pumping the body full of antibiotics, which kills off the majority of bacteria, leaving only behind those organisms which can survive, and then the body itself, under extreme stress, starting to remove nutrients from the gastrointestinal tract during recovery, seems to lead to certain bacteria becoming hypervirulent. They were once benign or hanging around. Now we've put enough selective pressure on the gastrointestinal environment that we've created environments where those microbes are incredibly pathogenic. So we're very interested in understanding that relationship and trying to track it down to see what kind of organisms might be able to be ameliorated in their stress response and prevent the development of surgical infections. I'd just like to finish by saying we do have a book out. Myself and Rob Knight wrote a book called Dirt is Good, which is a popular science book, which is a question and answer format encyclopedia almost, for providing easy to understand answers for many of your microbiome related questions, especially from pregnancy through 3 years, but it's relevant for all ages. And I'd like to stop there. Thank you very much.
Thank you, Dr. Gilbert, for that informative presentation. We will now start the Q&A portion of the webinar and will address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, can you tell us about the use of 16S versus shotgun metagenomics approaches in relation to the Earth Microbiome Project?
So absolutely. So the most important thing is we started the Earth Microbiome Project in 2010 when the cost of doing shotgun metagenomics was prohibitive. So we used amplicon sequencing in order to generate data that we could use to target environments for shotgun analysis. There are currently probably a little over 2,000 or 3,000 of the 100,000 samples, not very many, that have been used and had metagenomes generated. But that's changing dramatically. We're currently in the process of sequencing about 20,000 shotgun metagenomes from some different environments. And we hope by the end of next year to have increased that number significantly. And this is a lot due to the ability to multiplex metagenomic data on the Illumina sequencing platform, where it's been enabled us to get up to 50 or 60 metagenomes sequenced on a single run. And that allows us to significantly reduce the cost of generating that data. Obviously, those are shallow metagenomes, but the metagenomic data generated is still extremely valuable when generated at incredibly high volumes across many, many thousands of different samples, in enabling us to map functional variants in these different environments.
Great, thank you. Next one is, how do you see the Earth Microbiome Project fitting in with the One Health initiative?
We are at the Earth Microbiome Project, and my lab and my colleagues' labs specifically are an incredibly strong supporter of the One Health initiative. Providing an understanding of the factors, the components which drive our health, and link it to the health of environments, both agricultural and environmental, is incredibly important. So, as microbes are the bedrock of the biology of our planet, and interlink us very, very clearly with the environments around us, as I talked about in my presentation, microbial exposure from our environments is incredibly important in our health. We understand that to promote health, to promote wellness, it is essential to not just look at the microbes in our body, but to also look at the microbes that surround us and surround our bodies and that we are exposed to, and to try and promote the generation of environments which have beneficial microbial exposures at their heart.
Thank you. We have time for one more question. Can you talk about the relationship between asthma and the environment, and what are the latest findings?
So there are two camps here, and both of them are highly interrelated. But the central premise is that microbes in your body stimulate, or microbes you're exposed to as well, stimulate your immune system and alter your likelihood of developing asthma, and potentially alter the likelihood of exacerbating asthma attacks once you have the disease. So what we believe is that early life exposure, both respiratorily, so as I talked about in the Amish and Hutterites example, the children are breathing in those microbes, but also microbial health inside the gut of the child can play a very significant role in altering their immune state, in educating the immune system, and therefore reducing the likelihood that they get the buildup of sticky neutrophils or whatever other immune status actually influences that disease state. So Susan Lynch at the University of California, San Francisco, myself and many other people around the country are trying to tackle both of those angles and understand how they intersect to both treat and prevent the development of that disease.
Well, I would like to once again thank Dr. Gilbert for his presentation. I would also like to thank Illumina for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 13th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Bye.